This one?
I was wondering why that was so heavy. I'm on it. What are you doing, fool? Can you pick it up? I'm gonna roll over again.
She was here last week. She left school.
The lecture was really true to the post notes.
Well, I mean, it looks pretty. You get to work.
That somebody taught them that everybody's been doing for generation after generation. They don't really know what's going on there. But anyway, we're going to find out. So, food microbiology. There are some specific requirements, and oftentimes people want to know, what do we have to know? So here are the requirements for food microbiology. You need to know the difference between fermented foods and cultured foods. So I'll go over this and explain how they differ. You need to understand fermentation as a process at the molecular level. We're going to go over this in lecture in greater detail. So for the laboratory, there's kind of some basic ideas about fermentation that I want you to get without getting all the steps. So basically understand what's going on. You need to be able to recognize some potential health benefits associated with the consumption of cultured foods, depending on what stage they are. So we'll talk about those. You're going to be asked to recognize a variety of foods, specifically foods that we are making or that I'm demonstrating during this laboratory period. So there are a certain group of foods, and we use the term food very loosely because if it's just a beverage, we still consider that part of our food microbiology. You need to know the names of the organisms, microbial genera, because food microbiology is also going to be incorporated into laboratory exam 1. What you remember about species names, remember I said species names are extra credit. So the genus names are required. Species names are extra credit for food microbiology as well as the other species that we encountered earlier. Anyway, you need to know the names of the organisms that are involved in the fermentation that is associated with each food product. And you need to know what the fermentation products are that they're making. Yeah?
Are the species extra credit on the lecture exams too, or only one?
No,
okay.
Just on the laboratory exam. And then be familiar with some important foodborne pathogens. So hopefully I will remember all these things and make sure that you get a smattering of information as we go through this exercise. Okay, so first was the difference between fermented foods and cultured foods. Well, fermented foods contain fermentation products. That means that there were some microorganisms associated with the food. The microorganisms fermented the food. They produced an end product. Oftentimes it's acid, but sometimes it's alcohol. Could be some other type of substance. The organisms themselves are either removed or they're dead. If we're talking about something like sauerkraut, sauerkraut is a good example of a fermented food. The organisms that ferment are breaking down cabbage and they are producing lactic acid as a byproduct. Typically when sauerkraut is prepared for human consumption, it's cooked. Well, if you cook it, the bacteria are dead. But the fermentation product is still there, the lactic acid. So you get the benefit of the acid and the food is preserved. You can keep sauerkraut for a very long period of time. That's not to say that people don't sometimes eat fresh sauerkraut, in which case the organisms are still alive. Or at least some of them. Cultured foods also contain fermentation products, but by definition, if a food is a cultured food, it contains a live microbial culture at the time it is a finished product. So you expect them to have live microorganisms associated with them. Then again, if you put cheese on a pizza, cheese is an example of a cultured food, If you put cheese on a pizza and then put it in your oven, by the time you eat those organisms, they're not alive either. If you cook those guys, they're going to be dead. But cultured foods, as finished products, have live microbial cultures. So if we look at these images, the cultured foods are the ones in the middle. Cheese, a lot of different cheeses there, but yogurt is another example. So then another feature of cultured foods, they often involve milk and organisms associated with milk. The apple wine, which is on the left, and the sauerkraut that's on the right, those are fermented foods. Again, using the term food very loosely because apple wine is a drink. We just call it a food if you're going to consume it. So both of these are going to contain fermentation products, whatever the organisms produce as they're carrying out their metabolic pathways. So because fermentation is involved, whether we're talking cultured food or fermented food, we have to define what fermentation is. I think we did this in lecture because fermentation is one of the types of metabolism. Organisms are either fermentative or they're respiratory. And I think I gave you a kind of a brief definition. Anyway, fermentation is a metabolic process. A metabolic process means chemical reactions, right? And typically in here, what you're going to see is a series of events where you start with some kind of substrate, and then there will be an arrow to a next chemical, and then another arrow to the next chemical, and another arrow. Where the arrows are occurring, that represents some kind of enzymatic activity. So if you start with a substrate, the substrate gets converted into something, which then gets converted into something else and something else. You have this series of events, so that's a metabolic pathway. At the end, you have a fermentation product. The metabolic pathways that I'm going to show you tonight are attenuated. They're made short so you can see them, but usually these pathways are pretty long. They might contain 17 steps or something like that. Now, another way to define fermentation, this is the technical definition. Fermentation is the anaerobic decomposition of organic compounds, usually carbohydrates because they're the most abundant organic compounds. Anyway, anaerobic decomposition of organic compounds, and it's going to involve an organic compound as a final electron acceptor. I know I mentioned the electron acceptors to you guys before and said if you don't know what I'm talking about, don't worry about it yet. Eventually you'll have to understand, but I'm going to show you what I mean by electron acceptors here. So the organisms are breaking down carbohydrate, and in order to do this, they have to use enzymes. And very often those enzymes have some kind of helper. The helpers are often coenzymes, and the coenzymes can only function when they're in their oxidized state. How many of you are familiar with oxidation reduction reactions? A few. Okay. Some of you don't want to admit it. When molecules are reduced, they're carrying extra electrons. So if you have a coenzyme and the cell needs to use it in the oxidized state, and that molecule has been reduced, then it's got electrons on it, and the electrons have to be removed in order for that coenzyme to be used. So that's why we have to have a final electron acceptor to get the electrons off the coenzyme. I'll show you how in just a moment here. Anyway, so again, the organic compounds usually catabolize your carbohydrates. Carbohydrates are the most abundant, so we talked about carbohydrates, lipids, proteins, nucleic acids. Nucleic acids are a tiny amount of organic material. Most of the organic material is carbohydrate, so this is usually what they're breaking down. Consequently, when you look in biology textbooks, whether it's microbiology or general biology, when you start looking at catabolism, and fermentation is a part of that catabolism, what's being broken down is carbohydrate, and we typically start with glucose, and you'll see that happening over and over again. Fermentation as it occurs in eukaryotic cells often involves pyruvic acid as the final electron acceptor. So as I said, the electron acceptor is organic. It's actually made by the cell as part of their metabolic pathway. So it's an organic compound. They have it because they make it, and what it's going to do is pick up the extra electrons And again, you'll see more specifically how this works. It turns out pyruvic acid is not the only electron acceptor that cells can use. The other thing to think about is we're talking chemoheterotrophs here, organisms that are using the catabolism of organic compounds to get energy and carbon. When we first start talking about metabolism in lecture, we're mostly focusing on catabolism to release energy. We want to see how cells make ATP. But if they take organic compounds apart, they take something really big and they break it into smaller and smaller units, then those little units can be used to build new things, kind of like Legos. You can buy a Lego starship, take it all apart, and build a Lego yacht. Cells are doing that kind of thing all the time. So we're going to talk about both energy and carbon source. So chemoheterotrophs are breaking down organic compounds because they're going to use these as their source of energy and carbon, so they can build new parts, and they have the energy to do it. And here's a simplified version of fermentation, this little diagram. So this is showing glucose on the left, and then you see an arrow that indicates some chemical reaction is happening. This is a very short diagram because actually to get from glucose to pyruvic acid requires a whole bunch of steps. This is a short version of fermentation. In the process of cells converting glucose to pyruvic acid, they're going to release energy because glucose is a 6-carbon compound. Pyruvic acid is a 3-carbon compound. Notice you take this apart and you get 2 3-carbon compounds. Anytime you break covalent bonds, energy is released. And the cell is going to use that energy to make ATP. They're going to convert 2 ADP to 2 ATP. So that's energy capture. Again, they're chemoheterotrophs. They're using the breakdown of these organic compounds to release energy they can use to do work. And then the other thing that happens during this conversion of glucose to pyruvic acid is a coenzyme called nicotinamide adenine dinucleotide, NAD, gets converted to nicotinamide adenine dinucleotide in a reduced state. So it's got 2 extra electrons and 1 extra hydrogen, so it's NADH. In order to run this part of the pathway, to go from glucose to pyruvate, the cell needs NAD in the oxidized state. If it's got extra electrons on it, they have to get those electrons off. So what are you going to do with it? An analogy that I sometimes use is think spoons. You come into your kitchen, you have only so many spoons. Think of the spoons as coenzymes. If you come into the kitchen and all of your spoons already have peanut butter on them, it's like, oh man, I want to use a spoon, and I want to use the spoon to stir coffee. I don't want that peanut butter on there. So you have to get rid of it. You have to take it off. In this case, you have to take the peanut butter off the spoon and stick it someplace else. Bad analogy? Well, I used to talk about wheelbarrows, and then I decided students don't use wheelbarrows that much. Everybody uses spoons. Well, okay, so here's spoon clean, here's spoon with peanut butter. Now, a peanut butter covered spoon is going to pass the electrons to some electron acceptor, which happens to be pyruvic acid, pyruvate. When the electrons get transferred to this, pyruvic acid picks up 2 electrons and 2 hydrogen protons and becomes lactic acid. Again, the arrow indicates the direction of the events, and the NAD goes back to the oxidized state. No longer NADH, now it's NAD. Okay? That final electron acceptor in this event is pyruvate. It's organic. The cell made it by breaking down glucose, and then it's going to use it as an electron acceptor, and the pyruvate gets changed to lactate, lactic acid. Kind of makes sense? Kind of, sort of. Okay, anyway. This is saying the same thing. Pyruvic acid accepts 2 electrons, 2 hydrogen protons. It's going to become lactic acid, which in the diagram is lactate because it fits. It's easier to put it in there. The first portion of the pathway is glycolysis. In lecture, we're going to talk about glycolysis in a great deal more detail. Glycolysis is the catabolism of glucose to form 2 molecules of pyruvic acid. I think it takes, I don't know, a whole bunch of steps. It's not 1. I want to say 17, I'm not sure that's right, but anyway, a bunch of steps. The purpose, again, is to break covalent bonds, release energy that can be used to make ATP, high energy compound. The cell needs that to do whatever else it's going to do. Whoops. Go back. During the process, NAD, which is a coenzyme, is reduced. It's going to pick up electrons and become NADH plus H plus. The spoon gets peanut butter on it. If we want to use that spoon, which glycolysis requires, the coenzyme without the extra electrons, that means you have to get those electrons off of there. So what are you going to do with them? You can pass them to the electron acceptor, which is pyruvate. The pyruvate picks up the electrons and 2 protons and becomes lactic acid. Lactic acid is the fermentation product. So what is being fermented? Glucose. What is the final product? Lactic acid. It just so happens that lactic acid is something that we have learned to like. We like the flavor of lactic acid. If you have bacteria that produce only lactic acid, it's the only fermentation product they can make, we call them homofermentive. Homo means same. You give them something to ferment, they're going to make lactic acid. It's the only thing they can make. What that means is they only have certain enzymes, they can only run certain metabolic pathways. They're always going to produce lactic acid as an end product. We also call these lactic acid bacteria. So when I was going through and talking about the groups of bacteria, Lactococcus lactis, perfect example of homofermentative. They ferment, they're going to make lactic acid. Leuconostoc is another one. Some Streptococcus species tend to be homofermentative. So they produce just lactic acid. So Lactobacillus, I forgot about Lactobacillus. So Leuconostoc, Lactobacillus, Lactococcus, they are all lactic acid bacteria. They're homofermentative. But, so here's our food products: sourdough bread, yogurt, cheese, pickles, sourdough pancakes. What we think of as the sour flavor, the sour of sauerkraut, the sour in sharp cheddar cheese, the sour in yogurt, that's lactic acid. Do our cells ferment? We can also run a fermentation pathway, and when our cells ferment and produce lactic acid, it hurts. That stuff builds up in your muscle, and it hurts. If you're an exercise enthusiast, they'll tell you pain is good. It's like, nah, it hurts. But yes, our cells will ferment also. As far as I know, they're homofermentative. Well, okay, so glucose is catabolized during glycolysis. The glucose, in the case of making cheese or making yogurt, the glucose is coming from milk sugar, which is lactose. Lactose is a disaccharide, so it's a glucose-galactose combination. The cell takes that apart. Galactose and glucose are isomers, so they have the same molecular formula, but they don't have the same exact configuration. And glucose can also be made from starch, because starch is just a whole big long chain of glucose molecules. So you get a lot of glucose from starch. Now, is lactic acid the only fermentation product you personally are familiar with? I'll give you a hint. No. What's the fermentation product in that stuff? The fermentation product.
Alcohol.
Alcohol. You'd be surprised how many times I ask that on an exam and people can't answer it. And it's like, seriously? You know what's in beer? Come on. Okay? If that substance contained lactic acid as a fermentation product, I don't think it would be nearly as popular. Now people pick it up as like, oh, I'm having some pickle juice here. No, the fermentation product there is ethanol. And what makes the foam? Seriously, you guys? What makes the bubbles in champagne? Carbon dioxide. Wow. Yeah. Okay, so when you have microorganisms that are producing some other kinds of products, they have to be running a slightly different fermentation pathway. So here we see the pathway. we start with glucose and we go to pyruvate, but then after we leave pyruvate, the arrow shows 2 end products down at the end. They're not lactic acid. You're still going to get 2 ATP. And actually, what this diagram does not show you, pyruvic acid is not the final electron acceptor. Instead, there's another molecule called acetaldehyde. And acetaldehyde is what actually picks up the electrons from NADH. So acetaldehyde is the final electron acceptor, and you get ethanol and CO2 as byproducts. If organisms can form more than one fermentation product, and it isn't lactic acid, then we say they are heterofermented. Hetero means different. We're going to be starting Physiological Unknown 1 on Thursday. All of the organisms that we use that are gram-negative and fermentative are heterofermentative. And they can produce a wide variety of different types of substances. So not just alcohol and CO2. They make a variety of different types of acid. They can make hydrogen. They can make what's called acetomethylcarbinol or acetylene. Anyway, this is showing us a picture of Saccharomyces cerevisiae. Saccharomyces cerevisiae are the heterofermentative organisms associated with brewing. There are some other species of yeast that can be used for brewing, but oftentimes Saccharomyces cerevisiae is used. And I think I told you guys before, Saccharomyces means sugar fungus, and cerevisiae is cerveza. It's the sugar fungus used to make cerveza, beer. Now, what are you doing tonight? Well, one of the first things we're going to do is observe some curds and whey. How many of you have seen curds and whey? Okay, some of you have. So we're going to, I'm not sure exactly the best way. Right now, the substance is in the incubator. Usually what I try to do is put it on table 2, kind of in the middle, and then people can kind of gather around and hopefully see the curds and whey. So we're going to look at this. This is our cheese making operation. So in the laboratory manual, it talks about how cheese is made. Basically, you take some fresh milk, you put buttermilk in, which is the starter culture. So the buttermilk contains the organisms that are necessary to make cheese. And then you put in rennet. It's a proteolytic enzyme that will cause the milk protein to coagulate, to form a solid with any luck. Anyway, so you're going to get to observe that. Then you're going to work in table groups to prepare sauerkraut. We have 4 table groups down the middle, and each table group has 6 people. So you have 6 people at a table, and then we have a table group on that side and another table group on this side, but I want you guys to go with them on that side so that you're going to make that a group of 5. So that we have 5 groups, most of them groups of 6, and 1 group of 5 to make sauerkraut. And I'll give you specific instructions on the board. Then you're going to work in groups of 2 to 3 people. Probably it's better to work in the center bench. The center bench is 2 groups of 3 rather than 3 groups of 2. If you all divide yourselves up into groups of 2, we're going to run out of flasks. So do groups of 3, but you guys could be a group of 2 if you want to, because you're the only 2. Sometimes there's people missing. But here, if we miss people, we throw you guys into those open spaces. Anyway, so we're going to make some apple wine. Then you're going to work as a class to make root beer. This kind of happens on the side and on top of everything else that's going on. So we're going to make some root beer and bottle it up. We also have to label it so to make sure we keep track of our root beer, because invariably, come the semester, we get to sample root beer at the end of this laboratory, and they're always trying to give us root beer that somebody else made. I said, no, thank you. We want our own root beer, please. Anyway, step 5, each person is going to obtain a BCP lactose plate. BCP is brome cresyl purple. So it's a pH indicator. The medium is violet, purple. Each person is going to take one of those plates. What I want you to do is divide the plate in half. Just use your wire loop and cut the auger so that you've divided it into 2 sections. And then you're going to put cultured food yogurt on one side, streak some culture on from yogurt, and on the other side, buttermilk. And then when those have grown, we'll be able to look at some of the organisms associated with yogurt and cheese production. There's some information in the lab syllabus that talks about cheeses and different kinds of cheeses, and also the organisms associated with cheese. There are ripened cheese versus unripened cheese. So a ripened cheese, that term applies if the cheese is allowed to sit for a period of time after the initial processing steps. Oftentimes it's aged for weeks. Well, what is happening is the organisms that are going to be producing the fermentation product are given longer to do it. So they get to sit there and continue to ferment, and they make more and more of their product. Sometimes a ripened cheese will have a secondary organism type applied. Notice it says secondary flora. That's kind of old-fashioned lingo because fungi used to be considered plants. They're not. So it's really a secondary microbiota that has been added, fungi. So if you eat brie cheese, there's a mycelium on the surface. If you eat blue cheese, there's mycelium inside. The blue is penicillium. You're probably going to learn more about cheese than you ever wanted to know. An unripened cheese is a cheese that is a finished product at the end of the initial processing steps. Probably the best example is cottage cheese. Although the cheese that we're going to make in lab tonight, it's kind of scary in itself, it will be an unripened cheese. And if you care to sample it, you can. We'll be very careful about not allowing contaminants to get in there. There's also differentiation between soft cheese and hard cheese. Well, so a soft cheese is one that has 50 to 80% water. It's soft because it has a very high water content. When cheese is made, typically, again, you put milk and buttermilk or milk in a starter culture, and then some kind of enzyme to make the milk coagulate. You get curds, and then you separate them from the whey, which is the liquid behind it. If you want a hard cheese, then you put that into a press and press the water out of it. Depending on how much water you press out, you can have soft or semi-soft, or you can have hard or very hard. If the cheese is hard, then it has 40% or less water. Something like Parmesan, solid Parmesan is pretty hard. That's a good hard cheese. Again, it has less water. Now, the other thing that's shown in the lab manual is the organisms that were associated with cheese making. One of the most common is Lactococcus lactis. So Lactococcus lactis, and then there are two subspecies, Lactis and Cremoris. So Lactococcus lactis lactis, Lactococcus lactis cremoris are in many different types of cheese. They're not the only ones in there, but they are homofermentative and they're producing lactic acid. So you've all probably tried sharp cheddar versus mild cheddar, yes? So the sharp cheddar has a much more acidic flavor to it. That's because there's more lactic acid. Why do you suppose it's more expensive? Yeah, because it's aged longer. It takes longer to make a sharp cheddar because it's ripened and it's going to take a longer processing. You have to let it sit around. Or actually, I had a student that was a ranger on the Olympic Peninsula, and she said she used to go out on her ranger duties and she only bought mild cheese, and then she carried it around with her, and it fermented in her backpack or wherever she had it because it was warm, and it became sharp cheese. So you could do that. It'll continue to ferment if you warm it up. There's some information about yogurt. Typically, the organisms associated with yogurt production are Streptococcus thermophilus, and in microfood books, they call the Lactobacillus bulgaricus, but there's been a change in the classification, so now it's Lactobacillus delbrueckii bulgaricus. So bulgaricus is a subspecies. Somewhere in the lab manual, I've abbreviated to Lactobacillus D bulgaricus. Anyway, those organisms, when they're together, produce lactic acid and acetaldehyde. And there's enough acetaldehyde in yogurt that you can kind of taste it. The aldehydes have a nut-like flavor, so it's sour, but there's another little subtle flavor in yogurt, and that's acetaldehyde. Now, the yogurt that you get to sample tonight is yogurt that I made last night. I came over here to start Physiological Unknown 1 organisms, and Elaine reminded me, we're doing food micro tonight. I said, oh, okay, go home, make yogurt. So it's freshly made, and it's made using the recipe that's in the lab manual, if you are brave enough to try it. there's also mention in the laboratory manual about the potential health benefits of yogurt. One of the big benefits of making yogurt rather than purchasing it is it's cheaper because milk is less expensive than yogurt. But the other real benefit that I notice is most commercial yogurt has way too much sugar in it. Way too much. It's like, really? Come on, you can cut that sugar down significantly. So the recipe that's in the lab manual, basically you take milk and put some dry milk with it, which reduces the water content, and then you just mix that in there, and then bring it up to a temperature that's almost boiling but not quite. Put a lid on it and just let it sit for a period of time. When it has cooled down to about body temperature, then you mix some yogurt with it. And I usually use Mountain High yogurt vanilla. So it has just a little bit of vanilla flavor, and only the sugar that came in with about a cup of the Mountain High, this is going into about a quart of milk. So it has a really reduced sugar content. And then you just let the organisms do their fermenting thing. Well, okay, so they're going to ferment, and what they're doing is breaking down milk sugar, lactose. So one potential health benefit of eating yogurt, if you are lactose intolerant, a lot of the lactose is gone because the bacteria ate it. They broke it down. There's going to be some still left in there. But you get the protein benefit of milk without the problems associated with lactose. Also, the organisms that are alive in yogurt, if you make it at home, you get more of the organisms still in a functional state. And they're supposed to be good for your gut microbiota. I'm not sure how long they survive, but if you're taking antibiotics, one of the things you can do to counteract the effects of taking antibiotics is eat freshly made yogurt. It's also supposed to reduce serum cholesterol levels, but I don't know how. I don't know why it would, but supposedly it does. Now, there is some information about foodborne pathogens. There are organisms that are pathogens, actually cause infection. If you eat them, then they can infect, and they can go into your cells and cause a lot of damage. Escherichia coli is one of these. In the lecture syllabus, there's a couple of pages, and they're labeled the dark side of E. coli. And it talks about all these different strains of E. coli and what they can do. But they are actually invasive, so they can cause severe illness. Salmonella is another one. Those organisms are invasive, so they go into your cells. They can reproduce inside your cells and cause serious problems. Vibrio is another organism. There are different species of Vibrio. Of course, Vibrio cholera causes cholera. Vibrio cholerae causes cholera. But there's Vibrio parahemolyticus, usually associated with oysters. If you eat oysters raw, you can be infected with Vibrio parahemolyticus. And then there are organisms that cause intoxication. This is different. This means the organisms are producing toxic byproducts that if you ingest the toxins are going to make you sick. Probably the worst one is Clostridium botulinum. This became a problematic organism when people began to prepare food and can it, that is, put it in a closed container and seal it in there. Because Clostridium organisms are anaerobic. They're obligately anaerobic. So you seal them inside a container. They don't need oxygen to grow and reproduce. They grow in there and they produce the botulism toxin, which can cause complete flaccid paralysis. We'll talk about how that happens in lecture later on, but it can kill people. Primary reason is because paralysis of your skeletal muscle, not so good. You have to breathe. And breathing requires intercostal muscles and diaphragm. Those are skeletal muscles. If they don't work, you die. So botulism, potentially deadly. Another organism that produces toxins that can really make you sick is Staphylococcus aureus. So if you've heard about Staphylococcus food poisoning, it's because the Staphylococcus are not invasive, but they produce these toxins that cause vomiting and diarrhea, usually at the same time. Pretty nasty. Well, okay. You get to sample a variety of cultured foods. To make you feel better, these are purchased. They'll be in a cart. They'll be out in the hallway. So we don't bring those in here. But you have a variety of different cheeses. We usually have some crackers, some sourdough bread. Chocolate. Did you know chocolate is a fermentation product? Yeah. The fermentation happens in when the cocoa beans are in their pods. You're supposed to note the texture and flavors of the cheeses and look at the kinds of organisms that are associated. You're not necessarily memorizing all the different types of organisms that could be in cheese, because some of them are kind of weird.
Well, I have a question then. So is it when eating on cheese, lots of different flavors, some you might like, some you don't like. Is it the flavor of the bacteria?
So, good question. Sometimes the answer is yes. Some of it is the milk. So different types of milk can be used, and that will flavor it to some extent. Some cheeses have a lot more salt than others, but cheese like brie, there's an organism in there, Brevibacterium, and Limburger cheese has a different species of Brevibacterium. They produce flavor components that some people don't like.
Yeah,
to
like,
me,
I don't care for brie, but I like camembert.
right.
So does that mean I like the bacteria in camembert? I don't like.
Well, there's also fungus. There's mycelium on the outside on both of those. They could also be influencing the flavor. So it's a variety of different things. I'm not sure exactly what the difference is between those. Okay. I used to describe the flavor of brie as cow pasture. That's what it reminds me of. But you get used to it, and then it's like, ah, that's pretty good stuff. Or not. Anyway, the organism associated with cheese that you need to know is Lactococcus lactis. But then there's a bunch of different ones. There's bacteria, Propionibacteria in Swiss cheese that they produce propionic acid, which is another flavor component. So there's a lot of different things going on. You're supposed to record some information on the chart pages. That's again for your own use, because I never ask you guys to turn that in. But it's just to get you to think about cheese in a way that you probably never did before. And maybe you don't want to. What's growing in my cheese? Oh, that's the end. Okay, so I'll put some information on the board. Give you some more specific. And we want to save a little bit of time at the end to make sure that we have time to go out and enjoy our picnic, which is the sampling of food products. So you're going to be working in groups for most of this. It doesn't matter too much which of these things you do first. So sauerkraut. Okay, so for the sauerkraut, you're going to be working in table groups. What you're going to do is obtain sections of cabbage. I believe these are going to be cut and wrapped for you. You're going to use these relatively dull knives to cut the cabbage into very fine shreds. Please be careful, because you can cut yourself badly with dull knives. You're going to be cutting on cutting boards. You can put a piece of aluminum foil down on your board if you want to. Don't shred aluminum foil into your sauerkraut. So, before you cut this, weigh your cabbage. So you're going to weigh the cabbage first, and Breezy has set up our weigh. Where are you going to weigh? You're going to use the balance that's right here, so you come through this door and there's balance right there. The cabbage sections are going to be wrapped in aluminum foil, so you're going to weigh the cabbage plus foil. And then you're going to subtract the foil weight. So I'll put a little piece of foil on the balance to find out how much the bit of foil weighs, because you want to know what the weight of the cabbage is, because you have to add salt that's influenced by the weight of the cabbage. So you're going to weigh the cabbage, and then you're supposed to add a quantity of salt that's 3% of the cabbage weight. Okay, so to do that, you weigh out your cabbage, then you're going to multiply the cabbage weight by 0.03. That'll give you the amount of salt that you're going to use. So you weighed the cabbage, you weighed out the salt, then you're going to chop the cabbage up.
There's a screw protector on it.
Are you like just gonna put it on? So how are you feeling?
The freaking edges that get caught in my hair.
I can't overemphasize this. You need to cut that cabbage into fine shreds. You probably have some experience with sauerkraut, although some people really don't like sauerkraut, so try to stay away from it. I want you to make sauerkraut this evening, not containers full of fungus. This, basically what we're going to do is set up an enrichment. So you're going to chop the cabbage into very fine shreds, and I do mean fine, and then you're supposed to mix the salt. Some people recommend that you mix the salt in with your hands. Well, you can do that if you want to. Then you're adding unknown microorganisms from your skin. Some of them might get involved in the process. Doesn't matter too much, but you mix in the salt with those fine shreds, and then you set up an enrichment. Part of the enrichment has to do with the salt. If you put salt in there, what kind of an environment are you setting up? Salty, but there's another term for it. It has to do with tonicity. Hypertonic. And what's that going to do to the cells? It's going to make water come out of the cells. Then what's going to happen? So after you've mixed this up, when I say set up a microbial enrichment, put the cabbage in the larger of 2 beakers. So these are our cabbage fermentation chambers. There's 2 beakers, nested beakers. You're going to put the cabbage in the outside beaker, and you're going to put water in the inside beaker. Do not put water with your cabbage. If we've created a hypertonic environment, water's going to come out of the cabbage, and it's going to come up between the 2 beakers, and we're going to have an anaerobic environment. What kind of metabolism is going to happen in an anaerobic environment? Well, according to the information I'm giving you, and what we're talking about tonight. Fermentation, right? So what we're doing is setting up a situation. First of all, put the salt in, and you're kind of looking for halophiles, but then you're going to create this hypertonic environment. It's going to pull water out of the cabbage. The water will come up and form the brine, which then is going to make this environment anaerobic so that fermentation will take place. And as the fermentation happens, the organisms produce acid, and then they go through these population changes. Cabbage has lots of different organisms associated with it, lots of fungi. If you don't cut your cabbage fine enough, you won't get the brine, you won't get an anaerobic environment, and you will grow fungi. And it's just a mess. So don't do that. Make sure your cabbage is chopped fine. So you put the cabbage in the larger beaker, you put the weight, which is the water, in the smaller beaker on top. Make sure your beaks line up. Beat your beaks. Make sure they're in line. Maybe that's not how you spell it, but anyway. Beat your beaks. Make sure your beaks are in line. Oh, and then cover it. So then there are rubber bands in this box. The plastic wrap is here, cling wrap. You're going to make a piece of cling wrap. It's going to go over both beakers. You're going to tie it on there with a rubber band and then label it. Make sure you put a label on it. So label your setup. We're going to leave them sitting on the table, and at the end of the lab, we're going to check to see what the pH is in there. Hopefully we'll remember to do that. Okay, so that's the sauerkraut. So for the apple wine, again, you're going to be working in groups. Most of you are groups of 3. Only this side bench is a group of 2. You guys want to come back over here? Anyway, so in this case, you're going to put, well, so these are the flasks for the apple wine. They're 250 ml flasks. I wouldn't put more than 200 mils of juice. So somewhere between 150 and 200 mils. Again, the apple juice that we're using is commercial apple juice, so it's clean. Those flasks have been autoclaved. Notice that they have aluminum foil sticking up the top. These things have stoppers that have a piece of glass tubing coming through. The glass tubing extends down into the flask a little bit, which is why I don't want you to fill these things up. You have to have some space for the bubbles that are going to form. So you put your juice in there, and then you're going to add some yeast. You are not going to put your yeast directly into the apple juice. Instead, you're going to put your yeast into tubes of deionized water, sterile water. I don't know if it's a specific time that you're supposed to let those guys sit, and where the sterile water is. This is our sterile water. So these tubes have about 5 mils. They have about 3.5 mils. It really doesn't matter. You're going to put a little bit of yeast in here. Probably the most important thing is don't put too much yeast into the water. The yeast is dry powder, and when it gets into the water, it will tend to expand just a little bit. You want to allow it to hydrate in water before you put it into the juice. This is supposed to prevent the cell membranes from being damaged. So we hydrate it in water first. ultimately, we're gonna put our yeast water mixture into the juice. So when you add the yeast, put the tube between the palms of your hands and roll it to mix it. Then let it sit for about 5 minutes, and then add that to the juice. Now, you can add sugar to this if you want to. We used to say if you put sugar in, you were supposed to warm the juice to dissolve the sugar. Don't warm it, because it's not good for the yeast. Students tended to get their juice way too hot, and then their yeast would die, and that wasn't good. So, once you put the yeast in the juice, you can add sugar, but it's optional. The apple juice is pretty sweet, but if you really like a dessert wine, you can add a little bit of sugar. Just don't warm it up. And then you set up the gas trap. Basically, you have a flask. You have some juice in here. That flask has a stopper with a piece of glass poking through it. You're going to take off the aluminum foil and you're going to attach a piece of rubber tubing. This rubber tubing attaches to the glass from the top. So you're going to attach it and bring the tubing down on the side and put it inside a sterile glass tube. So this is a clean tube, not sterile water. So this is our sterile water. These are sterile tubes for gas traps. Don't mix them up. Well, we can't put yeast in tubes without water. Anyway, once you have this set up, put water in this thing. There's not enough water in these, so don't use these. You're going to put your own water in here. It's not going to go into the juice. It's just to allow CO2 to bubble out. Probably the best way to strap this on here is use white tape. Tape it on so it doesn't fall off. Okay? The root beer, as I said, we're going to work as a class group to do that. And the rest of this stuff, you're kind of on your own. You get to do the sampling and so forth. Oh, but the street plates. So the BCP lactose plates, one per person. What I want you to do, take a BCP lactose plate. They're very distinctive. They're purple. So the BCP lactose plates, very purple in color. You're going to take a loop and cut this thing. So you're dividing it in two parts, and then you're going to streak yogurt on one side and buttermilk on the other. What I'm going to do is set the plates. So we're going to have two plates, one that's labeled buttermilk, one that's labeled yogurt. I'll put those plates up here on the front bench. These guys can move over here. So we'll have this burner going. So you can come up here, take your sample, bring your loop up, put yogurt on, put buttermilk on, then take the plate back to your workstation and re-streak to spread this out. Okay? And then lab manual says to put this in your lab drawer. I'm thinking maybe what we'll do is put them in the anaerobic chamber instead. We'll put them all together. I don't wanna, if I put them in the incubator, I'm afraid they'll be in there too long. Even by Thursday, the color of the plate will change completely. But what we can do is put them in the anaerobic chamber. The organisms will grow a little bit better in there, and we'll just leave it at room temp. We'll gather them up. So beyond that, we have to make root beer. We have to taste a bunch of cheese. Okay, first thing we have to do is look at our curds and whey. So I'll get those out. So who's in the middle on table 2 here? Yeah. Where'd she go? Oh, can we move her stuff over? No, project, she'll come back, her stuff will be gone. What happened here? Well, we just moved you over. Sorry. I don't want to disinfect
Pause it.
I know, right?
Rennet is protease enzyme. It is supposed to cause the milk protein, casein, to solidify. This is always, it's like, did it work or did it not work? I never know. Oh my. So if you want to gather around, what you see is curd. So this container was full of milk, and now what it's full of is a curd and whey. So whey is spelled W-H-E-Y. It is the liquid portion of the milk once the curd is formed. This is why cheese is expensive. Most of the milk is water. It's whey. So you can see it's not very big. What we're going to do now is cut the whey into sections. You're supposed to cut it into little cubes.
So what you're cutting now, that's the actual whey?
This is the curd. The whey is this stuff. Now what you do with the whey is variable. Sometimes people use it for soup making, or they can use it to make bread, or they can feed it to their pigs, or they can throw the whey away.
You know that?
That's what we're going to do with it.
The protein?
Well, would anybody eat that? Because, like, you know
The
that,
whey?
Yeah, you know that one nursery rhyme, whatever. So
Miss
girl
Muffet?
eating her curds and whey.
Yeah, this is what Miss Muffet was having. She was having curds and whey.
It's just called cheese.
Okay? So there you can see the curd now. The curd is the solid part, the whey is the liquid. And now to continue the cheese processing, what's going to happen, well, I'm going to add a little bit of salt to this to make it a little more flavorful. And then I'm going to warm it gently, which is supposed to make the curd contract. And our goal is always to make squeaky cheese. Have you ever, any of you gone to the Tillamook cheese making?
No.
They have these little packages of squeaky cheese that you can get, and when you chew them, they squeak. So it has to do with the texture of the curd. But that's curd and whey. And again, you can see how much of the milk is liquid. That's not terribly useful for making cheese. So then you would pour this through cheesecloth, and the cheesecloth will separate the curd from the whey. And then, in this case, our cheese will be a finished product at that point. You can have curd on a cracker later if you want. You're brave. Okay? Everybody get to see curds and whey? More than you wanted to, probably. Alrighty.
So there's this type of protein called whey protein.